So hello everyone, welcome back to Hasvora Education. Today we are going to be revising all the practical stuff that you need for the paper 34 that is going to be held on 27th May of 2025. So this uh, revision guide is especially for that students, and today we are going to be revising the type of dilutions, the how to prepare the slide, how you are going to be writing the plan investigation question, and the type of the test that you essentially need to know. And in the next video, I'll try to make a full guide on the plan diagrams that you need to know. So now let's begin. So the type of dilutions and how to perform them. The first one that is mostly asked by the examiner is the serial dilution. So it is the general process of preparing a less concentrated solution from a solution of greater concentration. So sorry, I have written the name a bit wrong. It's not serial, it's simple. It's simple dilution. I just wrote it wrong. So it is the general process of preparing the less concentrated solution from a solution of a greater concentration. So we use the formula M1V1 is equals to M2V2 in this type of dilution. M1 is the concentration that you are being originally provided in the start, and V1 is that how much of the volume of the stock solution of the or the original solution that you are being provided, is that how much of that volume are you are going to be taking. So we keep this unknown, and M2 is the concentration you have to make. Like for example, if they are asking you to carry out a simple dilution, and for example you have a original concentration of 10%, then we know that we are going to take four more readings. So it can be 8%, 6%, 4%, and 2%. So this is the concentration that you have to make, and V2 is how much volume you have to prepare. For example, if you have been provided with 20 centimeter cube of 10% stock solution, and I have to make 8%, 6%, 4%, 2% each of each of 20 cm cube. So now this will be the V2. 20 cm cube will be the V2, and so this will give us the V1, which is how much we have to take from the original solution. For example, I have made these diagrams that I have been provided with 10% of 20 cm cube S solution, and then I have to make 8 cm cube, 6 cm cube, and 4 cm cube, each of 10 cm cube. Not 4 cm cube, it is these are the concentration 8%, 6%, 4%, each of 10 cm cube. So now I will apply M2V1, M1V1 is equal to M2V2. The M1 will be the 10%, which is the original concentration, and V1 will be the unknown, because we do not know how much we have to take. The M2 will then be the concentration I have to make, which is 8%, or it can be 6%, 4%, 2%, which ever you are going to be making. And V2 is the second volume that I'm going to be making. So this will give us V1, which is 8 cm cube. So it means we are going to be taking 8 cm cube from the original solution, and then how much of the solution we are going to be making. For example, if it's 10 cm cube, so we take the 8 cm cube from here, and then we add 2 cm cube of the water to it. So we will have 8% concentration of the solution. In the same case, we are going to be doing the same for the 6% solution, but we are just going to be replacing the 8% with 6%, and this is going to be giving us the V1 6 cm cube. So it means that we are going to be taking 6 cm cube from the original one, and then adding 4 cm cube to make it 10 cm cube. And then goes for the 4%, we are going to be taking the 4% instead of 6%. So it will give us the V1 as 4 cm cube, and then we are going to be adding 6 cm cube of water. Now the other type of dilution that we do is that the seal dilution. So it is multiple progressive dilution in which each concentration gets halved each time the dilution is performed. So for example, if I have a 10% of the solution in the start, I am going to perform the first dilution, so it is going to be making that 5%. Then I am going to make another dilution, it is going to be making it 2.5%. And then another, it is going to be making 1.25%, and then another, so it is going to be end up with 0.625%. So in this case, we are taking the half, or we are actually halving each of the concentration uh, progressively. So for example, I have been provided 20 cm cube of S with 10% concentration, and question states to perform Cl dilution. So let me clear a point that many of the students will be confused at how are we going to be knowing that whether we are going to be performing the Cl or the simple dilution. So the question is going to state it. If I have 20 cm cube of 10%, then halving each time results in 5%, 2.5%, 1.25%, and 0.625%. So, so I'm going to be taking the 20 cm cube of 10% S, and they often require to make require you to make a diagram. So I have also included the diagram of this example. For example, I have this 20 cm cube of 10% S. So I'm going to take 10 cm cube of 10% from this S, transfer it to another beaker, and then add equal amount of that of the solution of the which I have taken from the S. I'm going to add equal amount of water, so the concentration will half. And then from this beaker, for example, if I have taken from this to this, and then this becomes 5%. Now from this 5%, I'm going to be taking 10% to the another beaker, and then I'm going to do the same, and I'm going to transfer 10 cm cube of water. Now this becomes 2.5%. Now I'm going to take 10 cm cube of uh, of the 2.5% S to another beaker, and then I'm going to transfer 10 cm cube of water to it. So it becomes 1.25%. And then from the 1.25%, I'm going to take 10 cm cube of the 1.25% of S, and then add in the same manner I'm going to add 10 cm cube of water, so it gives me at the end 0.625%. So this all these beakers will have 10 cm cube in the end, except the 0.625%, because it will end up 20 cm cube, and there will be no more transfer from it. So in this dilution, we actually take the solution from the previous one. Like you can see that we at first we took from this one, then this one, then this one. While in the simple one, we take from the stock solution or the original solution that will be placed on a table in your practical area. Now let's come to the food test, which are the most common one. I have just you can say arranged it on the first one, the most common one, and then we going to the least common. So the first one is the Benedict's test, which is for the reducing sugar test. Now what are the reducing sugars which can carry out a process that is called reduction? So the sugars that are being you can say categorized in this category, the reducing sugars, that are fructose, galactose, glucose, the the maltose, and all type of the sugars except sucrose, and the polysaccharides like the glycogen, starch, and cellulose. So the polysaccharides and sucrose, these are not the reducing sugars. And this test actually gives us the colors in a, you can say, it varies as the time progresses. So the color is not always red. The color changes range from the green, orange, red, brown, brick red. So as they range from the green to the brick red, the concentration is actually more and more. So how do we perform this test? We have to add 2 cm cube of the sample solution or any volume that question states. More commonly they will state 2 cm cube, because it is more preferable when you are using a test tube. And then we have to add equal volume of the Benedict reagent. So when if we are taking 2 cm cube of the sample solution, then we are also going to add equal volume or 2 cm cube of the Benedict reagent. Then we are going to be heating it in a water bath for 4 to 5 minutes at 80 Celsius. If the solution turns brick red within the time, then it indicates the presence of the reducing sugar. Else, if it remains blue, then no reducing sugar is present. But it is not gonna just, you can say, just suddenly change to brick red. It will first turn a light green, then a bit orange, and then when you put it when the time progresses, it is going to be showing you a brick red color in the end. Now comes the non-reducing sugar test, which is done for the sucrose. It is a bit similar to the Benedict's test, but first we have to hydrolyze it so that we can check that is it a reducing sugar or a non-reducing sugar. So first we check if it gives negative test with Benedict reagent, or if the question mentions that it is non-reducing sugar or it is sucrose, then we know it for sure that it is a non-reducing sugar. So what are we going to be doing when we have confirmed that it is not it is a non-reducing sugar? We add 2 cm cube of HCl and add equal amount of the sample solution from it. And then we heat it for 4 to 5 minutes at 80 Celsius in a water bath. There is no change in this case, okay? It is going to be remaining as it is. Just we are just heating it so that it hydrolyzes, and there is no visible change when it hydrolyzes. So what happens as it hydrolyzes? The sucrose is going to be converting into an alpha glucose and a beta fructose. So then the alpha glucose is going to be giving you the Benedict test positive after it is being hydrolyzed. So when we heat it for 4 to 5 minutes at 80 Celsius in the water bath, then we wait for some time after we take the test tube out so that it becomes cool, like you can easily touch the bottom of it. So when the test tube is cooled, then we add the NaOH pellets to it, one or two in quantity, to the test tube so that it neutralizes the HCl that is being present and it does not just mess up our result. Then after that, we are going to be performing the Benedict test, and if it comes positive, that it means that it contained non-reducing sugar because we have hydrolyzed it, and if not, then no sugars will be present in it. The third type of test that we most commonly do is the starch test. We take 1 cm cube of the solution in the test tube, or you can also take 1 or 2 drops of it on a white tile if the question states, and then we add a drop or two of iodine onto it. If it turns blue black, it contains carbohydrate, else it don't contain. If it remains brown upon the addition of iodine, because it's just the color of iodine that is brown. The fourth type of test is the Biuret test. So we add 2 cm cube of sample solution to equal amount of NaOH, then add drop by drop copper sulfate solution. If it turns violet, protein is present, or if not, the blue color remains, then the protein is absent. So now most of you will be confused that we just write that add the sample solution to the Biuret reagent. There is nothing known as Biuret reagent, okay? The NaOH plus the copper sulfate is called the Biuret reagent, and you have to add it in this quantity. You have to add the sample solution and the NaOH in equal amount, and then add a few drops of copper sulfate solution to it. Now the fifth type of test is called the emulsion test, and that is done for the lipids. We add equal volume of the sample solution and the alcohol or ether, and then we add a little amount of water. I have just not written it. Add a little amount of water, and after doing this, what we do is that we shake it vigorously. If a white emulsion type appears on the top of it, then it indicates that the presence of the fat, else no fat are being present in the solution. Now let's come to the plan investigation question, that how you have to be writing the plan investigation question. It is the question, most of you will be just confused that what is the plan investigation. It is the question that you can see that it just mentions that this whole experiment has now been performed. Describe any modifications that you could perform for example, if we are just assessing the concentration of the glucose in the different type of the solution after performing the dilution, we are just assessing the concentration of the glucose in the different type of these solutions. So they are asking us that describe the modification that you would perform in order to have a better result of the solution which have the more amount of the uh, most amount of the concentration. So how do we do that? We take 3 to 4 values of independent variable. So what is the independent variable which we are changing? For example, in the case of the dilution, it is the concentration because we are changing that. We have to add 3 to 4 values of independent variable with units in our answer. So it can be the oxygen bubble, the concentration, the temperature, the pH. It can be anything that you are actually changing. State the constant you have been keeping. For example, it can be time. You are allowing all the solution to be in the water bath for the same amount of time. You are keeping the volume the same, the concentration. It also matters on the type of the question. Now, the third point that you have to be mentioning in your question is in your answer is how do you reach a conclusion? Like how would you conduct the experiment and how would you use that values to identify the trend? So, for example, in the most of the cases, like we do is that we draw a table and then we see that whether the increasing concentration increases the time taken for the color change to appear or not. So this is how you reach the conclusion. And then we add a measure to improve the result. For example, increase the sample size, perform the test 2 to 3 times with each of the concentration, take the average time, then use concentration with smaller gaps. Like for example, if you are using the Cl dilution, you know that Cl dilution just halves the concentration. So we could use a simple dilution to keep the concentration with smaller graduation or smaller gaps. For example, 10, 8, 6, 4, 2 rather than 10, 5, 2.5, 1.25, and use a more precise instrument. For example, if you are measuring the color change when the color changes when you are measuring the time. You could also use a colorimeter, so that the type of the precision that you are increasing by the instrument. Now you could also include the hazards that are being associated in the experiment and their protection. For example, the high type and the high risk hazard is the injury or it can be a chemical. The medium level risk are the stains, the irritation, the corrosive chemical. It can be dilute chemical like HCl, and the low one can be the methylene blue and the starch solution because they are a bit corrosive. And the precautions can also be included, like for example, if you are using uh, a needle or you can say a type of knife to cut up the pieces of onion or something, the layer of the epidermal cell, then you could use a finger clip if you are using the blade or knife, and you could also use the goggles if you are handling chemical, gloves, or you could also use lab coat. So this is all the this is all the variables or the points you have to uh, just ensure that you include in your answer when you are answering a plan investigation question. Now let's move on to to how to prepare the slide. For example, it is very rare, like I have just seen it 2 to 3 times in 10 years, that they ask us to prepare a slide of the onion epidermis after they have put it in the different concentration of solution, and they are actually requiring us to see the plasmolysis or the turgidity of the cells after it has been taken out of the solution. So how do we prepare that slide? For example, I'm just assuming that it is the onion epidermis because it has been coming 2 to 3 times in the last 10 years. So we use a pipette to add few drops of water first to the microscopic slide that we have been provided. Then we peel the thin layer that is on the top of the onion section of the onion epidermis using a forceps, and we just cut a small piece of it with the help of help of a blade, and then with the help of the forceps we lay it flat on the slide. Then we add 2 to 3 drops of iodine to stain it so that we can see it easily with the microscope, and then we carefully apply the cover slip using the forceps and needle to avoid trapping of the air bubbles because it will just distort the view that you will have under the microscope. Then we blot excess of the solution with the tissue because it will cause the cover slip to we just move. So we are going to be blotting the excess solution with the tissue paper or the blotting paper, which is kind of like white paper that is used to blot the paper, the solution. So this was all the stuff that you needed to know for the practical paper, and I'll be making a next video on the plan diagrams together with their slides that you need to know for the, and it will contain all type of the slides, all type of the transverse sections and longitudinal sections that you need to know. So stay tuned for that. Like, share, and subscribe. Share with anyone that you think it will help. I'll meet you in the next video. Till then, goodbye.